So welcome back, Klausians. It was a real pleasure to exchange with many of you at lunch. I'm Thomas Gervais. I'm a professor of engineering physics at École Polytechnique in Montreal, Canada. And thanks again, Klaus, for organizing this. I say just 1 minute. Just the one thing that strikes me about the community of Klausians is how many different fields Klaus has pioneered through his career. And I'm in the microfluidics/bio period of his lab, but we meet people, CVD, multi-scale modeling, purely theory, very applied, machine learning, pharma, and that's really something that inspires me as a professor. And if I have 2 fields at the end of my career, I'll be really, really proud. So that's really, really an impressive achievement. Before I introduce the speakers, all of them need no introduction, actually. I just have to say to everyone that we will have a picture after. So a family picture up front here. So please do not leave this room before the picture is taken. Okay, so this will happen at the end of this session. And I looked through LinkedIn at the various speakers and found that, well, my session is maybe the title of it should be something like Industry and Innovation, on how science has a second life after the publication in Klaus's lab. So we will hear from many people who are accomplished in industry or in making startups or participating in technological innovation. And the first one of them actually requires absolutely no introduction in this community. I will not even start to state all his achievements, and just please welcome Professor Bob Langer.
Well, thank you so much. It's really a pleasure to be here, and congratulations to Klaus and Chris. And I've had the pleasure, as I looked at this, of now writing 16 papers with Klaus. I will mention a couple later. Having him as my boss, as when he was department head, and even getting involved in companies with him, which you'll hear from the next speaker. But at any rate, I thought I'd start it by telling you a little bit about my own path, which probably is very, very different than almost any chemical engineer. So I got my degree here, actually at MIT in 1974, and all my friends then that year pretty much went into the oil industry, and I wasn't that excited about it. So I actually, when I was supposed to be doing my PhD thesis, I ended up spending most of my time starting this school for disadvantaged high school kids in Cambridge, and I got involved in creating new chemistry curricula, and I really liked that. So one day I saw an ad by the way, I did get 20 job offers from oil companies. Not that I was that good, but I got 4 from Exxon alone. But I just wasn't excited about it. But anyhow, I'm saying that because you'll hear some of the other job experiences. But so at any rate, so I really liked that. So I saw this ad to be assistant professor of chemistry education at City College of New York. I wrote them a letter applying for a job because they wanted development of chemistry curricula, but they never wrote me back. So, but I liked the idea. I found about 40 ads to do something like that. I wrote to all those colleges, a lot of them not particularly good ones, but it didn't matter. None of them wrote me back. So that wasn't going so well. So I started to think, what other way could I use my chemical engineering education to help people? And I thought about medicine. So I wrote to a lot of hospitals and medical schools, and they didn't write me back either. But one day, one of the people mentioned to me, the surgeon, Judah Folkman, was at Children's Hospital in Boston, and they said sometimes he hires unusual people. So I wrote him, and he was nice enough to offer me a job. And his idea was if you could stop blood vessels, you might be able to stop cancer. And it was very controversial. Biggest problem, though, was that there was no way to study blood vessels at the time, so we had to invent one. And our idea was to use certain places, like in the body or chick eggs, where there were hardly any vessels or none at all. Like in this case, it's the rabbit eye. And we'd put certain tumors in. And then we needed a tiny delivery system that could deliver large molecules. All the molecules we were isolating were large. And no one had ever done that before. And there was hardly any work actually done in that area either, except for one company, Alza, in California. And Dr. Folkman was on their board. So he went out there and asked them, could they help us? But they all said, well, that's impossible. You could never develop a tiny delivery system that could deliver large molecules. But if we were going to solve the blood vessel problem, we had to solve this one. So I spent 2 years working on it, experimenting with many different techniques. And I found actually over 200 different ways to get this to not work. But eventually I was able to make these tiny little particles. You see a picture of one, another cut in half. They're like nano or microparticles. And we published in the journal Nature that you could use this for the first time to deliver these large molecules. It was also the first time that nucleic acids were ever delivered from tiny particles. But what happened was when I was pretty excited about this, but what happened is when I presented the work, people really didn't believe it. They'd say that the molecules were too big to get out, like you can't walk through a wall like that. And the solvents we used, they said they'd destroy everything. And the consequence of that for me was that even though we, published these papers, that my first 9 grants were rejected, and I applied to chemical engineering departments then after 3 years as a postdoc. A lot of my friends said you shouldn't do that forever, but anyhow, I liked being a postdoc. But I started applying to chemical engineering departments, and nobody would hire me. They all said this bio stuff I had in mind didn't make any sense. So I actually got a job in a nutrition department here. I got it, I think, because Nevin Scrimshaw, who was the department head, was kind of a benevolent dictator kind of person. So he liked me and he offered me a job, but he didn't ask anybody else in the department what they thought, which would have been okay, except for one thing, which the year after I joined, he left. So a lot of the faculty told me I ought to leave too. So this is actually a quote from Mike Marletta, who was my colleague then, and I'll just read you what he wrote. This is referring back to 1979. He said, one evening he went to a faculty dinner with me and some senior MIT professors. He said, a senior scientist sat quizzing us while smoking a cigar. He said, when the older scientist heard my concepts for drug delivery, he blew a cloud of smoke in my face and said, you better start looking for another job. And so over the years, I was in that department, and then actually MIT got rid of that department in 1988, and everybody was looking for a job again. So I ended up coming back into the chemical engineering department. But actually, to be blunt, a lot of people really, not only these guys, weren't so nice to me. But I was very lucky. Klavs came around the same time, and he along with the Stephanopoulos and Bob Brown, were very nice. And that meant a lot to both me and my wife, that, to have that kind of support. Well, here's Klavs. And actually, over the years, fortunately, some of the things that I started doing that people didn't think were very good, well, they did lead to things. So one of them, the blood vessel inhibitors, it took 28 years from a paper we wrote in Science, but starting in 2004, what you see is Avastin gets approved, which is the number 2 or 3 best-selling biotech drug in history. And now, many of these are approved. Probably people estimate they'll end up affecting over 500 million people. And they're not only the only drugs that, they're not only used for cancer, all kinds of cancer, but they're really the only drugs that can prevent macular degeneration, which is a leading cause of blindness, or diabetic retinopathy. And the drug delivery work, I got involved in starting companies and licensing. That also led to a lot of products, which are shown here, everything from, different, like drug-eluting stents to ways to treat brain cancer, prostate cancer, prevent opioid addiction, and so forth. But I thought I'd tell you a little bit more about one of the companies we started. So I kept doing work in microparticles and nanoparticles, and in 1990, I mean, I didn't know any of this, but in 1990, there was a paper in Nature showing you could use messenger RNA to get some biologic effects, but it caused some safety problems. So what happened was, 15 years later, Caitlin Carrico and Drew Weissman at Penn developed a way to modify the mRNA so it wouldn't cause those effects. They actually got the Nobel Prize for it last year. They wanted to start a company, but nobody would give them money. So they couldn't do that. But about 5 years later, another fellow came to see me at Harvard, and he also was doing this, building on their work. And so and he wanted to start a company too, and he started talking to me about this, and but Third Rock and other places didn't give him any money. But I thought that the big reason, to my opinion, was people felt if you injected mRNA, it would just get destroyed. But we had done all this nanoparticle work, and I thought maybe that could make a difference. We'd shown that you could deliver it when people told us it was impossible. We actually also came up with coatings that allowed it to last for a long time. We published that in Science. We came up with other types of lipids that would, make it pretty harmless. And Dan Anderson, who was one of my postdocs, did an amazing job, and really up till the time he was involved, there were maybe only 30 or 40 lipids, and he had this idea of making libraries and making thousands of lipids so you could start choosing what you wanted. So I thought Dan was great, and and actually UMass, so he was, even though he was a postdoc with me, UMass offered him a full professor job, several $100,000 and a giant startup package. But I felt it would be much better if he stayed here. So we talked to all kinds of people. Nobody would hire him. He's a molecular biologist. But fortunately, we introduced him to Klavs and Arup Chakrabarty, and they hired him jointly between IMES and the chemical engineering department. Klavs was head of the department at that time. Dan, by the way, amongst other things, started CRISPR Therapeutics, which just a few weeks ago developed the very first CRISPR, a way to treat sickle cell anemia. So, it's probably a good investment. On Moderna, just so you know, this is what Moderna did with the COVID vaccine. It just totally changed history, really, that you could, in 2 days, design this and then start clinical trials, within 2 months. Nonetheless, the criticism for me doesn't stop. This is the Boston Globe just a few months later, the front page headline saying, even after we announced the results at Moderna, that it wasn't how you do science. Of course, Boston Globe isn't usually right. But and ultimately, of course, as people know, this one got approved and is used all over the world, affecting millions of lives. And not only that, I think you'll see it affecting millions more. This is the idea that you can use it to treat cancer. It's what's called a personalized cancer vaccine. And it's done exactly the same way you do the COVID vaccine. This is for melanoma, and it's now in phase 3 trials, but you select these mutations, and you, in this case, what patients got Keytruda. That's an immunotherapy drug. It's probably the best one around. It's Merck's drug. Moderna did this with Merck. And it's always a double-blind trial. Half get Keytruda, half get Keytruda plus this vaccine. But if you look at the data, it's pretty remarkable. At 2 years, the risk of recurrence or death is reduced by 44%. 3 years, reduced by 49%. Metastasis, which I didn't put up here, reduced by 69%. We also came up with other ideas as I started doing work. One of them was that you could create new tissues and organs. Just to give you an example of this, one of them is artificial skin, which you can see here. But just to show you what it can do, if you had a baby who was burned like this, you can put this on them. And what happens is that this is 3 weeks later, and this is 6 months later. And these are now approved by the FDA for people who are burned or have diabetic skin ulcers. So, and now, of course, what's exciting is lots of people all over the world are working on this field, not only to make new tissues and organs, but even organs and tissues on a chip. So, so with Klaus, I also, in addition to having him be my department head, we published, as I mentioned, a number of papers with him, 16. I won't talk about all of them, but this was an early one that Jörg Lahan, who's now professor at Michigan, did with us, where we modified surfaces. And of course, you're going to hear more about this in a second, but I was really glad. It must have been about 2009. Klaus called me up and said, we should write a grant to NIH, and we did, on the idea that maybe we could do use some of his microfluidic work to insert things into cells. And so with Armand, who's going to be the next speaker, we came up with a way to do that. And I think it's really a revolutionary method. And we actually published quite a few other papers in this area too. So I'll just show maybe one or two more pictures. Yeah, this is, I guess, the three of us a few years ago. Armin was kind enough to give me that. And, so and it really was a great, I think, experience for all three of us to participate in this discovery, which was published in PNAS, Nature, and other places, and even, as Armin will go over, now has led to several companies that he's run. So I thought I would just summarize by just really thanking Klavs. I feel both as a friend, as a colleague, as a department head, as a collaborator, it's been wonderful to know you and to work with you. And really, when I was asked to speak today, I absolutely wouldn't have missed it. And really, congratulations and thank you for being the person you are.
You surprised me. Actually, I'm delighted that I don't have to interrupt Professor Langer with the sorry 1 minute. So thank you for keeping on time. Any questions for Professor Langer? All right, let's thank him again. Right, our next speaker is Armand Sharai, who did his PhD co-advised by Bob Langer and Professor Jensen, and then went on to make several startups, one of which is Squeeze Biotech, for which he was CEO for 10 years approximately, and now he's CEO of Portal. So let's hear about his journey.
Thank you. Definitely an honor to speak for Klaus's, what'd you call it? Post-tenure party. So I'm one of the traders from the biotech side instead of academia, hence I forgot you got to wear a sport coat to these things. But my first, this clicker doesn't work for that. So my first interaction with Klaus was when he was the intimidating sounding department chair, and I was a clueless recent ChemE undergrad coming in. And he did set my stereotype for what Danish people are like, because I had never met one before. So forever in my head, it's someone with nice hair, very soft-spoken, very nice, but then that silence is deafening when you're an undergrad that has no idea what to say. And in our first meeting going through the projects, I definitely remember Klaus going through a litany of chemistry projects. And as some of my fellow PhD and postdoc lab mates from the time can attest, even though I love them, I would fall asleep instantly when it was a chemistry talk in group meeting. So I was trying to be interested in the chemistry aspects of what Klaus was talking about. And then in the last minute or so of that meeting, he says, oh yeah, we have this project where we shoot cells. I'm like, what? So Andrea, who's also going to speak later, and Klaus, two immigrants from Italy and Denmark, had really embraced American culture and decided to solve problems by shooting at things. So what they were working on was a gun to fire materials into cells, because cells are really good at excluding whatever they don't want inside them. So me being childish, I was like, this sounds awesome. We're going to work on a gun where you have cells going down a channel and they're being fired at, such that you can deliver the material inside them. And this being a serious problem in biology, it seemed like as good a way as any to do that. So I signed up. We were off to the races. I had a tendency to hire undergrads to do the real work, so I could analyze data and chase after them to do their work. For some reason, I like that. But we had a little team of undergrads going, and Klaus was very supportive throughout those days. And I had learned lessons from Bob's stories about writing letters that don't get replies. Turns out if you CC Klaus and Bob, everyone will respond to you. So getting collaborators was very easy if you just CC'd them. If you didn't CC them, good luck. But CCing them was a key ingredient. So we got going on this project. And like most grad school projects, when they get pitched to you by their PI, they don't really work, but you think they do. So we were having a lot of trouble shooting at cells. You would think it would be easy. And one problem was that if you saw that design earlier, as the cells would go by, if you actually run the fluid dynamic simulations, you need an insanely high pressure jet of fluid to be able to penetrate into that zone. And so one of the problems was the pressure from that jet would dissipate too quickly, and the cells would probably just like barely bend away, and nothing would happen. So that was really confusing us. And what we ended up doing is adding in this constriction element to really jam the cells up against the gun and shoot them in the face so that hopefully it would start working. And this project started to work. We thought we had the gun working because we were really squishing the cells up against the gun as we fired at them. But the parameter space made no sense. So if the gun was set to high or low, it didn't matter, which was confusing. So one day we turned the gun off and the whole thing still worked. Turned out just squishing cells at high speeds was enough to disrupt their outer membrane. And so that became the basis of our squeezing discovery that Bob was alluding to, where the process could be boiled down to you had a cell, you would mix it with whatever cargo you're trying to deliver, and then run it through a constriction element at high speeds. That rapid mechanical deformation of the cell would disrupt the outer membrane, and it meant that anything that's outside can then diffuse into the cell. And then the cells, interestingly, have highly conserved membrane repair mechanisms, so they detect that disruption, they close it up within about 30 seconds, and your material stays inside. And what was exciting about this was that it seemed very broad in its capabilities. So we were able to do a lot of immune cells, stem cells, all kinds of different cell lines. So many cell types that were conventionally extremely difficult to deal with. And because it's just a diffusion-based mechanism, it appeared you could put whatever you want into these cells. It didn't matter if it was a nucleic acid or a protein or a peptide. It's just diffusion. Anything you want goes into the cells. The other interesting aspect was that you seem to keep the cells pretty normal on the other side. So you wouldn't get dysfunction in these cells, and that meant collectively that now you could engineer many different things about a cell biologically to create all kinds of different potential cell therapies or ask interesting biological questions. The other nice thing was that because it was such a simple concept, it's very easy to scale it out just by adding more channels in parallel. So the clinical versions of the chip would have almost 1,000 channels in parallel, and that way you could get 10 billion cells a minute through. To give you a sense, a typical patient, the number of white blood cells you can pull out of them is on the order of 2 to 10 billion. So you could do a whole patient's worth of immune cells within a minute, which is extremely fast for the kinds of things you would want to do in a clinical setting. And the way this was distinguishing from what people had done in the past is that things in the past were not that great, hence disparaging cartoons. But if you were trying to do viruses, there was a lot of constraints on what you could package into a virus, what cell types the viruses were capable of transducing, and they can be insanely expensive at scale. Another common technique is electroporation, where people will expose cells to an electric field and try to drive material into them using that electric field. I don't know if you like getting electrocuted, but cells apparently don't. And so this would cause a lot of dysfunction issues within the cells, and there were also cargo constraints as well. Like there are certain cargoes you can't really deliver in this way. But with Squeeze, it looked like you could basically put whatever you want into whatever cell type and do it in a relatively simple, streamlined process. And so we decided to start a company around this. Given that we were a ragtag band of undergrads, this is us posing with John Harvard's statue. I'm not in this, I had nothing to do with it. But we had to go through a maturation process to be presentable to investors and others, so you can see we cleaned up pretty well eventually. And from there, we were off to the races, having no clue what we were doing. But with people like Klaus and Bob, you could get a few doors to open at least to get you started. And over the years, we had an exciting journey with Squeeze, which was the first generation of this. So like Bob was kind of pointing out too, we got some pretty nice recognition in the field for trying to solve this very esoteric but fundamental problem to how you can understand and engineer cells. We started to work on the therapeutic side of this, and Roche became one of our big collaborators over the years. Towards the peak of Squeeze, we were about 150 people, and we had 3 clinical trials going, so I'll give a bit of a preview of what we were up to there, where one of our main theses was around how do you activate a patient's own T-cell response against a tumor. So this is what we were partnered with Roche on. And the way this works is you take various immune cells out of the blood, you mix them with a cargo that is going to be representative of the tumor, so a peptide or a protein from the tumor that's going to be your antigen of interest. With our process, it was pretty simple to deliver the materials, and then once you have these squeezed APCs, they now contain the antigen in their cytoplasm going back into the patient. And what these APCs are meant to do inside the person is to present the antigen on what's called MHC class 1, which is the receptor that your killer T cells look at to see what they should target on a potential tumor cell or an infected cell. So the idea was you're kind of cheating and telling the T cells exactly what to look for, circumventing all of the natural processes that you normally rely on to do this, and then be able to drive a powerful anti-tumor response. And this is just one example of the things that these kind of mechanical delivery technologies would help open up, because you had so much flexibility on the cell type and cargo that you could work with. You could now engineer virtually anything you want about these cells. On the business highlights side, one exciting, very memorable thing was that we IPO'd on the NYSE back in 2020. One cool thing they do is they light up the entire floor with your logo, so that was pretty nice to see, and this was all day, which was kind of crazy. This was during COVID, so unfortunately, Klaus and Bob didn't get to go because they had restrictions, but it was pretty cool to see. And for the clinical programs we were working on, there's a whole range of oncology work that we were doing, so there were 3 different clinical trials there, but there was also applications that we had worked on over the years in autoimmunity, for example, particularly going after celiac disease and type 1 diabetes. So these mechanisms I mentioned where you can instruct T cells on what to attack, you can flip them and instruct T cells on what not to attack. So for an autoimmune disease, you could use mirrors of these mechanisms to shut down immune responses instead of turn them on. And a lot of the immune responses you work on for cancer are also very relevant to infectious diseases. So we were looking at things like hepatitis B, and also for my postdoc, we had a history of some work on HIV for these applications. Now, one of the most exciting things about doing this was actually making a difference for a patient. So we had a few clinical responders during our trials, which was definitely some of the most memorable moments along the way. And one of the other important things that we had been able to show is that you could actually make a cell therapy in under 24 hours, which was pretty unprecedented. Even still, no one's there yet, because it currently takes weeks to do this, and that's part of what makes cell therapies cost hundreds of thousands of dollars to administer. It's not the pharma making a lot of money off of it. They're actually losing money usually on cell therapies and trying to make it work. And so with our newer company Portal, we're focused on continuing this journey to eventually get towards point of care production, where if you can really simplify what it takes to engineer a cell from a patient, can you boil it down into a machine that could be at the hospital, at the bedside, that can engineer these cells for you. And the 2 parts we're focused on is how do you have a kind of Intel inside, or we call it powered by Portal system, that could be integrated into all kinds of different equipment to enable these kinds of workflows, and then also get into a little bit more of the cargo side. So what are the mixtures of molecules you would want to put into a cell to dial in different functionalities? Because I would say you can kind of think of a cell as a machine, and the molecules you put in are the software programs that can reprogram what it does so that you can have the desired impact on the patient. Because the bigger picture motivation for us as to why we're trying to solve these problems in cell therapy is that some of the people in this room have contributed to going towards scalable chemistries, making a lot of these drugs possible for people. So the reason over 100 million patients a year just in the US will get small molecule drugs, or still millions will get peptide drugs and biologics, is that people figured out ways to do this in a scalable, cost-efficient manner. Whereas cell therapies are still at this phase where they haven't had their moment where, for example, someone figured out recombinant DNA and made biologics feasible, or people figured out scalable reactions and made it feasible. And we think by having these integrated engineering solutions for cell modification, that's what can unleash this world of possibilities by having the next transition in therapeutics be leveraging your cells as the main therapeutic engine, instead of small molecules and biologics. For those of you in small molecules, don't worry. We have a while to go. We won't ruin anything for you anytime soon. And just I wanted to end a note on, as we continue to try to make Klaus as read as possible at the end of each of our talks, I would say some of the most memorable things with Klaus was he was always willing to bet on his students. You could tell I was probably the insubordinate, problematic one at times, but Klaus would just smile and be like, I told you not to do that, but you did it anyway. And we'll just, we'll let you do it again, and you keep screwing this up, I'll kill you. All with a smile. So no, but really being willing to bet on his students over and over again, because without that backing, I would have never dared jump to the company side. Sometimes he has impractical advice, like when I was thinking about going to the company side. I'm like, how am I going to have time to do that? He's like, well, you could clone yourself and then do it. I'm like, okay, no. And also the patience through all the turmoil, because there's a lot of ups and downs with these things. And one thing that when things go wrong, you see a lot of real personalities come out, especially when there's kind of big dollars and big events at stake. But Klaus always remained kind of unselfish and committed to that bigger picture and that bigger impact, whereas you can see others falter when it comes to these things. So that's definitely a big credit to Klaus. And with that, like everyone here, I just want to say thanks again. And yeah, happy to take any questions if there's time.
Thank you, Armin. Any questions? Yes, here.
So after doing this, you inject it back into the patient.
Yes.
And what is the half-time? And is there an immune response to that?
Yeah, so, yeah. Oh, sorry, this isn't connected. Yeah, there's multiple dimensions to kind of the timing kinetics. This antigen presentation event is on the order of 24 to 48 hours. And then the subsequent T cell activation and proliferation tends to be on the scale of days. The key thing, though, is if you do this part right, your immune system is pretty amazing at forming memory. So even though the proliferative effects were only for a few days, if you get memory cells, you're protected as if this was your childhood vaccine. So it could be a decade later, and these immune responses will still be there. And a lot of this will just come down to the nuance of how did this activation event occur? And so one of the other things I've oversimplified this. One of the other things we were engineering is what other signals do those T cells see that would help bias them to a memory state? Just showing them the antigen alone is not it. You need to show them other things. Yeah. Good.
Yes.
You could comment more on some of the challenges with loading small molecules using this platform?
It works really well for loading small molecules, and actually with our new company Portal, as I've admitted, I'm suck at being a chemistry person, even though my degree says ChemE. A lot of people have been interested in using this for heterobifunctionals and other large-ish small molecules to put them into cells, because it's just a diffusion mechanism. So as long as the molecule isn't very hydrophobic or positively charged, which would make it stick to the cell membrane, it looks like we can deliver whatever we want to cells.
Last question.
The business case between selling the instrument for doing this versus kind of sounds like you're kind of going after developing whole therapies based on it?
I could go on for hours on that topic. The short answer is, so with Squeeze, we started off doing tools. The conundrum you face with doing tools is that from a VC business model perspective, they like high-risk, high-reward bets. Like they want to place 10 bets and have one of them exit for $1 billion plus. That's how it works. Placing 10 bets where each of them have a good shot at being a 20 million exit doesn't cut it. You get into this really weird zone where you might have uptake of your tool, which is what we had, but all the pressures say just go do the therapeutic, go for the big win. So with Squeeze, that's what we're doing. Actually, with Portal, we've kind of inverted things, and we just do instrument and consumables. We don't do the therapeutics ourselves.
Thank you, Armin, for sharing your path with us. So our next speaker is John McMullen, who's a senior principal scientist at Merck, and I believe John arrived in the days where I left Klaus's group, so 2005 until 2010. So looking forward to your talk, John.
So thank you for the introduction. Klaus, thank you for organizing this, inviting me. It's a real privilege to be here today and speak on some of the stories from my journey. I wanted to start this family reunion story with the slide that I was fortunate enough to share at AICHE last year at a session honoring Klaus, really speaking to all the dimensions that make him truly unique. And I wanted to use this opportunity to speak on behalf of everyone that, you know, as a student, you raised expectations for us for what we could do, and you gave us the tools, the guidance, and the patience to do that, to be successful both in research and beyond, so thank you very much. Now, the title of this talk is actually, Wait, You Can Do That in Flow, because that's sort of the energy that we get when we show our chemistry colleagues some of the things that we traditionally can't do in batch that we now can do in flow chemistry. It's also going to be a story about, you know, how we've leveraged the research that Klaus does in the labs into an industrial setting that allows us to provide medicine for people. Part of that is bringing disruptive technology into manufacturing, and there's been a lot of really smart people that write books about that. They compare it to crossing a chasm or how a single match lights a fire. When you're in the trenches doing it, it's more like pushing a wall, right? You're trying to get, you leave Klaus's group, you have all this innovation, you have all these great ideas, you're trying to get them used in an application, and for one reason or another, it doesn't always work out. And so it feels like you're just pushing up, but what you're really doing is trying to find the soft spot in the wall, right? You just need that one opportunity to break through and convince folks that this is a sustainable technology for innovation. And so I want to share that approach, that story that I've had at Merck, but it's true for everyone that's left Klaus's group. It's true for every industry that they've gone into. For drug substance, you know, we have any pharmaceutical company, their drug substance organization, the mentality when you bring an active pharmaceutical ingredient to the market is that the best science to build your molecule has to have the best chemistry at its core. There's more dimensions to it, like logistics and operations, but you cannot deliver the best science without the best chemistry. So this is an example of the synthesis used to make doravirine. It's an HIV therapy. This is what we would consider best chemistry. An overview of what this looks like is you have an ester that reacts with a diene and an aldol formation that then goes through elimination to get to this diene. Oh, well, shoot. So it's not showing up, but it's the dienes in the middle, and then you have this cyclization to get a pyridone, which then goes through alkylation to actually get to the API. Now, there's a chemistry design problem here, right? This little R on the starting material over here is something that you can actually play with. And what our chemist colleagues found is that by increasing the sterics of that ester, you can increase your assay yield in this reaction. The challenge, though, is when you carry it downstream to make this pyridone, the bulkier esters lead to product inhibition. So you get great assay yield at this point, but it doesn't translate down through the isolation into the thing. Thanks. Still helps out. Thank you. So we decided to go with the ethyl route, but this had a new challenge. The ethyl ester, the one on the top, is unstable. So when you're doing this in a batch reaction, it begins to degrade while you're producing it. And so this is an opportunity to work in flow where we can maintain cooler temperatures and short reaction times to maintain our product. And so this was our desire, which gave us this opportunity to redefine the expression from what we have used to a new one, which is the best reaction engineering brings the best chemistry to manufacturing. I'm fortunate that Steve Buchwald is no longer here. So we're just going to take that as gospel. This is what it looked like in 2013. And this was our first modern day continuous process at Merck. And so you have your starting materials in one vessel that are then pumped through. They meet the base, which in this application was potassium tertamylate. They go through a mixer, a plug flow reactor, and then undergo elimination in this downstream vessel. So that's the drawing in real life. It's Klavs height tall, and it's just made from tube-in-tube heat exchangers and reactors. So how did this first application go? I actually found the note that I sent to a leader in an email. Overall, the first production demonstration offered glimpses of future successes. And what happened was, what you're seeing here is is good enough as any illustration to show you. Oh, sorry, Klaus. Is this instability in the temperature readouts post heat exchangers. You see the red line is the base solution increasing up in temperature as it's going into this reactor, which is increasing the overall temperature in the reaction there. These spikes up are also corresponding to pressure spikes in the flow system, which is essentially telling you that you have clogs and blockage happening in your reactor. And so not only is this not giving you desired performance, but you have to shut it down, clean it up, and restart it each time. It doesn't take a Professor Jensen to figure out what the problem was. The base solution that we used in lab is shown here. It's very clean, and in the pilot plant, it's more like a slurry, right? So now we had a new challenge. We were able to go one step forward to demonstrate it, and then maybe 2 steps back to actually do it. And so now we had 2 challenges. One was to fix this problem to ensure that we had stable operations, and the other one was to now demonstrate more convincingly that we could scale it up and take it anywhere to manufacturing. So we applied the KFJ methodology in industrial research on these 2 problems. We first looked at the instability in flow. We were able to do experiments and show that the slurry is caused by the hydrolysis of the alkoxide, right? And so this makes essentially KOH. It's insoluble in the organics, and it leads to gelling more than anything. This is a thermodynamic problem, so we look for a thermodynamic solution. And essentially, we were able to show that if you just use a little bit of THF as a co-solvent, you can enhance the solubility even when there is hydrolysis. So just a little bit of THF went a long way here, and what you're seeing now is the next, jumping ahead, the next campaign that we did in the pilot plant where you're seeing stable temperatures the entire time. So there were no clogs, no excursions, and everything ran stable. Now, when we're thinking about scaling up, we also took another chapter out of Klaus's book. We're looking to go from this little mixer, which has a frit in there, that's what actually promotes the mixing, to pipe, where you're just relying on momentum for the mixing. So these are 2 different types of mixing, but their thought was the same. If we could characterize the Reynolds or fluid dynamics regime, we should be able to scale from one to the other. And so there's this really nice 1970s paper about how to characterize Reynolds numbers through a frit, and the point of transition to turbulences were a Reynolds number of 1 in these systems. So essentially, if we can just, what we showed is in the lab, if we operate at that Reynolds number of 1, the reaction performance, which you're seeing as red for start material and purple for product, gets us to a plateau where we're getting the desired performance that we want. So we just have to operate just a little bit past transition as we scale up. And so to get to the punch, you know, this was something that we've demonstrated both in the pilot plant. We transferred it over to a manufacturing place, and we're still getting, we're still matching our improved lab yield of 72%, and we don't shut it down. In the pilot plant, we did this to do 200 kgs of drug intermediate, which at the time was one of the largest demonstrations for a pharmaceutical company. But more than that, it was the breakthrough, right? It gave everyone confidence. We built new reactors. We demonstrated how you would run it in a GMP setting. We qualified it. We tech transferred it to a manufacturer. And so this now opened up opportunities that really have improved the way that we make molecules. And so these are more examples of just fast chemistry, but now we're going past the point where we're designing our own mixers with 3D printers, we're characterizing them, we're fabricating them to do more. That's opened up the opportunities to build more reactors and allow us to integrate with other technologies that we've invested in heavily, such as biocatalysis. So for example, this is a packed bed reactor in flow where we're essentially doing a transformation of a byproduct of cellulose to make drug substance intermediates, which is really nice from a sustainability perspective. When you do that type of modification, people then ask, well, could you build a reactor that allows me to do, say, photochemistry? And so this is actually technically an award-winning synthesis that allows us to produce belzutifan using photochemistry to process around another 200 kgs of drug substance intermediate over a 2-day period. And again, this just encourages our chemists to do more innovation. So now we're starting to look at electrochemistry and essentially the future of what can be done for these syntheses. Now, these are chemistry examples that are making kilos, but Klaus has also shown that you can use flow reactors to generate information. And so we've just talked about some of the reasons why you might do flow over batch from a chemical synthesis perspective, but when you start to do characterization and data investigation and kinetics, right, to do those experiments in flow, you traditionally would run at steady state, allow 3 to 5 reactor volumes to go to waste, take a sample, change conditions, and do it again. And you do that multiple times over multiple days to build a reaction curve that looks like this. You change conditions and you can get more. Now the way that you can do it in batch is significantly more straightforward. You just put automated sampler technology in there and you collect the profile for one experiment. So how do you maintain the benefits of flow for synthesis, but improve the way that you do data collection? And Klaus really spearheaded this approach that uses dynamic flow operations, where instead of operating at steady state, you're changing the flow rate in a controlled manner and monitoring the output as it's coming out. So let me just walk through a little bit here. So this is a schematic, shows you what a typical flow reaction looks like in the lab, where you have pumps that are going at their flow rates to set the residence time through the reactor. And essentially in a given experiment, your total flow rate and your total residence time are due to the summations here. So if you were to collect certain aliquots as they're coming out of the reactor, that would correspond to these data points for these steady state conditions here. Now, in dynamic mode, rather than operating the pumps at one constant rate, you program them to have a controlled profile, sort of like an HPLC gradient here. These now have a total flow rate that looks like this, so the residence time now is ramping linearly in this case because it's a 1/x relationship. And so as you're collecting aliquots at this point, you're actually getting them over a course of different residence times. And so this is a really nice data-rich experimentation approach to build reaction profiles and flow. Now, as you can imagine, the conditions that you set at the inlet are not actually the residence time that the fluid elements see as they go forward. And so to get around that, Klaus has shown that you just have to do a little bit of math that relates the flow rate to the volume and the times that you enter and leave the reactor so that you can convert the instantaneous residence time that is at the inlet to what the segment of flow actually sees as you collect it for analysis. So this was something that we used on a drug substance program that went back to that immobilized biocatalysis approach that was operating at 3 hours residence time. So you can imagine at 3 hours residence time, your whole day is just waiting for that one sample. Here what we did was we did that ramp and collected it over the course of around, I think, 10 minutes to 150 minutes. And this is essentially a month of data in just one day. And you can look at some of the metrics here in terms of the amount of grams, the materials that we were saving here. And so this is something that we've been very excited about to try into new things. And again, this is another breakthrough, right? Now we're looking at how to do more with that type of system, and if you put a PAT measurement at the outlet and an optimization control algorithm, you can do online optimization in dynamic mode. This has behooved us to go into the pilot plant and update our flow systems to have a feedback controller as opposed to just relying on steady state measurements. And now we're taking this same approach into new operations by looking at doing controlled crystallizations in batch mode. And so again, these are just things that just set off. And so, you know, reflecting again with a little bit of age and experience, right? Going backwards, it wasn't actually pushed on a wall, right? I had nothing to do with a lot of this. Way back from this wall is more like dominoes. And so this is Klaus pushing the technology ahead gradually such that as it gets, you know, vetted in the academics, we get an opportunity to apply it in industry. So thank you very much, Klaus. With that, if there's any time for questions, I'm happy to take them.
Thank you, John, for a great talk. We have time for one question. Anyone? So maybe when you I'm interested about this hole in the wall, how yourself coming to Merck with these new ideas, what was the moment where you could actually breach that and go people like, oh, this is okay, let's go, let's change the way we do things.
The aldol chemistry was a really good example. Flow, that was definitely it. Because
Maybe you can speak in the yeah.
yeah. So the aldol chemistry was a really good one. In general, I think that everyone's very excited to see new technology in the lab, but to actually demonstrate it in production, inclusive of even the logistics of like a batch sheet, the documentation and all of that, that I think is what requires people to really have comfort in actually putting it into their compounds. And I think that's true. Photochemistry was another example. We just happen to use it in flow. And I'm looking forward to like seeing new things like solid phase peptide synthesis and other ways to build these molecules. It's those moments when they actually get scaled up, when adoption happens.
Yeah, okay. Thanks a lot. So our last speaker before the picture, let's not forget the picture, is Dr. Andrea Adamo, whom I had the pleasure to work with and had spent many lunches and parties with him because we were at the same time in Klaus's group. He was a postdoc and a research associate, and now he's CEO of Zeitput Flow. And I already have one question: how did you come up with this name? So
I will close with that question. So first of all, thank you so much, Klaus, for the invitation to speak. It's truly an honor, and it feels a little bit a family reunion here. So there's a special feeling to this event, which I think it's a beautiful thing. So I want to start with a little bit of the history of flow chemistry, or I should say some critical milestones in the flow chemistry activities that run in the group. So everything, I guess, starts with microchemistry applications, small-scale reactors. And then there's a very big project that is the MIT Novartis event. So there's several faculties involved there. But it's really important in the history of the field in general because it's the first time that there is a full demonstration of an end-to-end production of a single pharmaceutical. So in the microchemistry, we are in the tens or hundreds of microliters per minute. We go 2 orders of magnitude. I guess here you were working in the 40 to 80 milliliters per minute. And it is a quantum step ahead. The next project that comes in line is the pharmacy on demand. The demonstration that you can do this eventually led to this program sponsored by DARPA. There's a few parameters that change. There is still an end-to-end type of manufacturing, but now the flow rates are a little bit lower. You are in the few milliliters per minute, up to 10, but you have to be able to synthesize multiple drugs as selected by the user. So now another challenge that gets added in the picture. And now after that, there's 2 branches, I would say, at least that's how I feel it. Perhaps Klaus has a different perspective actually on this. One direction goes towards back to desktop size synthesizer to explore a little bit more the interaction between machine and computers and how automation comes into play to go to higher functionality. So the MOD was designed to be able to optimize multiple multi-step reactions where the steps, it's up to 5 process steps that can be defined by the user. And eventually that led to the ASCO's project, in which the interaction with the machine, with the computer, gets taken to the next step of complexity. On the other side, the POD continues to grow. There's a POD2, I think there's a POD3, GMP demonstration. There's a variety of multifacets. It's essentially is to show that the technology is viable, the technology can be used for multiple campaigns in a reliable way and provide a full, I guess, a full demonstration of those capabilities. There's companies that come out from this effort. One is Zyput. So myself, I was involved in the POD and the MOD. When the ASCO started, I was on my way out. The minimal intervention on the POD2. Dale Thomas took those projects from the design standpoint. I was around when the MIT Novartis project was around, so I actually absorbed a lot of that knowledge. My Tide takes ideas and things, concepts that were developed on the ASCO's project in Klavs' lab, and On Demand took on the effort onto the Pharmacy on Demand. So this is it's truly an incredible journey. And while I was in the group, everything seemed so logic. You know, you have a chip, and then you have a pump. Now you control the pump with the PC. It seems so straightforward. And so I went on, I started Zyput essentially to commercialize equipment, because the effort that I did in POD and MOD, I really liked it and made it my career, really. Once I stepped out, I realized, though, that I was living in a world that didn't exist. Let me phrase it another way. In the Klavs Jensen group, I had lived in the future. So I stepped out, and I was sure that continuous manufacturing was the future, but nobody else believed that. So what I learned is that, indeed, everything was very rational, made sense, but it wasn't the future. With the experience I have now, so I travel a lot for commercial reasons. I attend a lot of flow chemistry conferences. And what I see on and on is that things that we have seen, you and I have seen in the group 10 years, 5 years, 15 years, 20 years ago, they're still not even reality in the marketplace. And so there is no question that the future is continuous, I think, for pharmaceutical. At least that's my opinion. But it's going to take 20, 30 years so that all these ideas that are laid out in this board and all this fantastic effort that was done will come to full fruition in the commercial marketplace. So this innovation is truly incredible. I think with these ideas here, we'll live for the next 40 years. People, so we have talked about science, we've talked about technology, but I think everything, you know, at the end of the day, starts with people and ends with people. So this is Klaus getting gowned up to get in TRL. I think now TRL is different. And there's a few pictures of people. There was a challenge in finding pictures, so there wasn't a specific intention to highlight somebody or leave somebody else outside. It's just that indexing of all pictures is actually not sophisticated, so it's really hard to find. But you can recognize some of the people that are here in the audience as well. And so, as far as myself, I went on to commercialize a separation technology that I developed in the Klavs lab during the POD process. Essentially is a way to enable inline liquid-liquid extraction. So you mix in a line and then you first separate continuously. And so as a company, we took on the effort of scaling up across 5 orders of magnitude. So you start with the lab, you can go to plant, and now the latest is 40 liters per minute. The reason is really what I call hybrid applications. I think is actually a very likely future in the next few years where you mix batch and continuous for hybrid systems, because it's, you know, if you have a driver from the financial standpoint to justify a continuous tool, then you will add it to your batch plant. The conversion is going to happen, but in my opinion, it's 20 years, 30 years to really get there. We arranged those systems in multi-stage configurations at different scales. Now we are moving into other unit operation. We have continuous hydrogenation. As much as I wanted to talk about it today, because it's very new and there's a number of new ideas in there for scalability and safety of operation, I actually picked a different topic because it has deeper roots in the Klavs lab. I must say, though, that this is really this design is a combination of things that I've seen in the group, just arranged perhaps in a new way. And so that takes me to the topic that I prepared for today, that is continuous liquid-liquid partition chromatography. And so when it comes to purification in pharmaceuticals, crystallization is the go-to technology for at least for more than 90% of APIs. Sometimes it's unsuitable, and so people will look at chromatographic-based techniques. And so HPLC is common knowledge, so I won't cover that. I'll just spend a few words on partition chromatography. So in chromatography, you have your sample that goes over the solid phase, and the chromatographic effect allows you to separate your analytes. Now, let's say that your solid phase, which we call actually stationary phase, is a liquid as opposed to a solid. Then if you have a sample, there I have a sample with red particles and blue particles, and I present it to a stationary phase, in this case would be the bottom green one, you get partition. Liquid-liquid extraction essentially between the two phases. And so if now you are able to take the moving phase, the yellow, and move it forward to a new stationary phase, you're going to have this phenomenon again. And so as you continue to cascade this, what's going to happen is that eventually you are sieving for the analyte that has greatest affinity for the yellow liquid, and the one that is affinity for the green liquid, so the stationary phase, elutes later. And so actually there was a Nobel Prize given for this in the early 50s, but of course the technology has the challenge of having a second liquidary phase that you keep as stationary. And so people have devised all sorts of ideas. The one that has been a little bit more common, perhaps in the last 20 years, is to use a rotor, to use centrifugal force to push your stationary phase towards the wall of the rotor. There are some applications in natural products. You see them a little bit here and there, but it's been not certainly mainstream. However, once you have a hammer, a lot of things are looking like a nail, so we have technologies for continuous extraction. So we started asking ourselves, what other ways can this be applied? And so if you think of an ideal separation, the ideal chromatography, let's say you have rabbits and turtles, and they're both running towards the right, and you have a treadmill that pushes things towards the left. So now you can find a way to tune the speed so that actually you can separate the rabbit from the turtle. And if you feed rabbits and turtles continuously, then you're going to separate rabbits and turtles continuously. And so that's what we want to do. And so we started asking ourselves if the membrane-based continuous separation technology that we have allow us to get closer to this type of concept. So I need a kind of side background in order to close the loop later. So the idea of continuous chromatography with a solid stationary phase is not new. It's been around probably more than 30 years with industrial applications. Essentially, you have different beds, different packets of solid support, and by playing with the position where you have your feed and the outlets, you mock up a continuous solution. The reason I have the slide is because this type of approach really allows you to separate rabbits and turtles. However, if you have a multi-peak chromatogram, a bunch of peaks are your turtles, and the other bunch is your rabbits. And so now let's go back to the idea of using liquid-liquid extraction and see if we can actually enable a truly continuous paradigm. So what you want is to feed somewhere, and then you're going to have a carrier, then organic and an aqueous that you feed from either side, and you have to play with the affinity that your analyte will have with either the organic or the aqueous. So you have to play with the partition coefficient in order to try to replicate this phenomenon. And so in a sense, it boils down to the way you would actually design the process. So what is key to have identified rabbits and turtles is really the control of the partition coefficient. You want one analyte ideally to have partition coefficient greater than 1, meaning it has greater affinity for the organic, and one that has it below 1, so it has greater affinity for the aqueous. And so what's drawn up here, this slide is a little busy, but you kind of need this to conceptualize the full thing. What we have, it's really a multi-stage countercurrent system that by designing a slightly different way, your extraction process actually approximates a truly continuous partition chromatography case. And so, as equipment manufacturers, of course, we're a bit more interested or excited by the equipment side of things. However, in order to come to fruition, we had to come up with a methodology on how to design such a process. And so, the first step would be optimizing your partition coefficients, and I'll close that at the end. What we did in the beginning was to take papers where people have done it with partition chromatography examples. We developed a model that's essentially a mass balance based model, so that actually you can predict or have a first guess understanding of the performance of the system and where you want to put your feeds and your relative flow rates. And then we can actually use Zyput devices to carry out lab experimentation. The key advantage here is that everything is miniaturized, so the amount of liquid, the volume of the device, the holdup is minimal, and so you can carry this out in the lab with small amounts of material. And then by using the bigger brothers of the same devices, you can actually take it to production. And so in the testing, we took a few cases. This is the separation of seringaldehyde from hydroxybenzoic acids and vanillinic acid. And the difference of KDs is good enough to get separation. And so when we run it into the system, we do get pure or fairly pure, 99.3% seringaldehyde pure, and we get rid of the other two components. We did another example with separation on enantiomers. This was interesting for us to explore, having in mind cases that could be more interesting in the pharmaceutical space. So propanolol, I think it's a beta blocker of first generation, so it's been around for a long time. There's a right and left molecule, and in this case, we use an enantiomeric selector, a chiral selector taken from literature. And so what happens is that this chiral selector binds to one of the two enantiomers, and so skews the partition coefficient. And so we were also able there to replicate results that people have obtained in the partition chromatography literature. We did a final exercise with different molecules, cheaper ones in this case, but to demonstrate that we were able to run it at scale. And so to close up, the motivation here was really to try to expand the scope of application of what we do. The motivation from an industrial standpoint is the much lower OPEX, but more importantly, the fact that you need to buy expensive solid stationary phase. So actually, the saving is estimated to be in the 60 to 80% range. That takes me to the end of what I have prepared for today. There's one last slide. I failed to address the issue of optimization of the partition coefficient. So we have this internal R&D effort in which we have built a machine to automatically create samples and measure the partition coefficient, so I can do it brute force. The motivation is the fact that solvation and partitioning are very hard to predict, and hence a machine should help alleviate that. Actually, this is a project that we're looking for collaborators to take it to the next level. And with this, I certainly, the time I spent in the Klavs Jensen group has shaped the rest of my career. I think probably I am the person that has spent the longest time in the Jensen group. And so it's here fairly noticeable because I think I know, or I've interacted with at least half of the people that are in this room. And in all honesty, when I was there, I gotta get out. Now that I look back, I said, thank you, Lord, that I didn't get out, actually, because I learned so much that it was worth it. And thank you so much.
Thank you, Andrea. One question. Yes.
What do you think is the reason why the continuous process is not allowed as opposed to batch? Why do you think it is?
You mean in general chemical processing?
ahead?
Yes, the reason is the following. So when you look at things from an industrial perspective, you have R&D, but then you have to go to production. So if you already have assets that are paid for, there's very little incentive. All your workforce is trained in batch, and the manager that makes a call is taking a personal career risk, so to speak. So the incentives have to be very objective. So you have to be able to demonstrate either an ROI or a technical superiority. And you would do that realistically in discrete fashion. So like what John said, John's story, I think is a perfect representation of what happens in the industry. You need the winning cases in order to convince all the surroundings. And so it's just a matter of time. It's like, I think it's more when as opposed to if. The yield? It depends on the case. Generally speaking, you get very good efficiency because in flow-based reactions, you have accurate control on engineering knobs. And so you do get better performance in general.
Thanks a lot. Oh, one more, why not?
Why Zyput?
So Zyput, that's a great question. So when I was a child, I used to make a lot of names, make up names in my games. And at some point, that trait left, actually. It started again when I had young children. And one of these names, you know, in every game, make 50 names during a day, and they all disappear, except a few. And Zyput was one that stayed through my teenage years, and I started a joke with my brother that if I ever had a business, I would have called it that way. So now 15, 20 years almost go by, and I have to name the business, and then I went back to that old story. But to add to your question, I'll take another 30 seconds. So I looked for a lawyer to register the name of the company, and it was obvious that this person didn't like the name. And so he asked me 3 times. And at that point, I was absolutely sure that it was a good name, because taste is personal, but I needed it to stick.
Great story. All right, so it's time for the picture. So we'll let Kathy take over, and maybe so we bring it. We all come forward, I guess. What kind of background do you want?
My name's Ryan Hartman. I'm with New York University, and I'll be the moderator of the last session of the day. And I just want to thank Klaus for allowing me to be a part of this special reunion. When I reflect on how Klaus impacted my life, he took a chance on me. I came back from industry to be an academic, but he really instilled in me the philosophy of applying fundamentals to discover new fields, but also at the same time that in academia, everything should be about the students. So this slate of speakers that I have, or that we have lined up, if you wanted to call this session, I suppose, computer-aided discovery of catalysis and reaction engineering, I think that would be fitting, but also the direction that Klaus has taken the field in recent years. And so I'd like to invite Bill Green to the stand, who's the director of the MIT Energy Initiative.
Talk about the, I guess, most recent part of Klaus's career, machine learning, and for what we've done on trying to predict properties of molecules and reactions. First, I'll give some history for those of you who haven't been around in recent times. So I joined the faculty 27 years ago. Klaus was already here. We were colleagues for a long time until maybe 2012. We'd served in each other's thesis committees. We helped each other's students, but really we didn't do any direct projects together. And then in 2012, I became the executive officer and Klaus was a department head. And so we worked together quite closely. And at that time, we renovated Building 66, and we had some other troubles, and we had a lot of things to deal with, and we worked together a lot. During that time, I don't know how he did it. When he was department head, he was still running quite large research programs. And one of the things that happened was they formed he had a project from DARPA, I think, to try to make a little box that could make a pill, and you just decide what medicine you wanted to make, and it would just do the whole thing. It would synthesize it from simple chemicals, and then it would make actually crystallize it and mix up with, I don't know, binders and make an actual pill. All in this little box maybe the size of this lectern. That was pretty show-off gizmo. So that was pretty cool. And so then DARPA said, but the problem with this box is you actually still need a human expert because you need somebody to tell the robot and the machinery what to do. So he said, okay, let's get rid of that human expert. So Klaus bravely decided that he was going to try to make the replicator from Star Trek. So if you guys know that, you go to the replicator, you say what you want. I want a hot cocoa. It gives you a hot cocoa. You say, I want a plush toy for my kid. It makes a plush toy for your kid. Whatever you want. So he wanted to do the same thing for chemicals. That you go in, just draw your molecule on the screen, and then you go press the button, and then out comes that chemical. This was pretty outrageous. But Klaus was DARPA likes pretty outrageous stuff. And so Klaus was okay with it, and he managed to sweet talk 2 computer science professors into agreeing to join this project. And he persuaded me too. He's very persuasive sometimes. I remember the initial conversations we had when we were putting the proposal together. It was like the Tower of Babel, because the computer scientists were speaking computer science language, and they had never seen chemistry since sophomore year of high school. And the organic chemists were with us, and they sort of a little bit understood the engineers, but had absolutely no idea what the computer scientists were saying. I remember the big shock when it came out that molecules can be cyclic. That completely threw the computer scientists for a loop, because they were used to linear programs. So anyway, but we survived, and we actually got funded. I think based on Klaus's excellent previous work, because obviously it wasn't really that we knew what we were doing. And so then we started to work. And then Klaus made a really smart move, was he recruited Connor Coley, who was an incoming graduate student, to agree to work on this project. And Connor didn't know enough to know how hard it would be. And so then he went and just took all the computer science AI courses and a bunch of organic chemistry and other stuff. And in 2016, Connor actually figured out how to do some of this. And so in 2017, we published 3 papers that made quite a splash in the pharma community. The pharma community was already getting a little bit fired up about restarting its interest in computer-aided organic synthesis. And then seeing these nice papers coming out of MIT made them really fired up. And so both DARPA said, oh, wow, you guys are doing so great on getting the computer to figure out the synthesis and direct the robot how to make it. We'll even give you another grant, and now we want you to do all of chemistry. So basically, the human won't even tell you what molecule to make. The human's just going to set specifications. I want a molecule that does this and this and this and doesn't do that, and computer, go to it. And so we applied for that one too. Klaus, I think, had some trepidation at the time. It was maybe considering early retirement. But anyway, he decided to stick with it, and we did it. And the pharma companies were going wild. And so Klaus caught that energy and organized the Machine Learning for Pharmaceutical Discovery and Synthesis Consortium, which was great. It tied us to what the pharma companies really wanted and really needed. And it also provided a lot of funding. And so between that and the DARPA projects, we were able to form quite a strong team of computer scientists, chemists, chemical engineers, all kinds of people. And then, when you have a big enough team, you get a lot of cross-fertilization between the smart students here at MIT. And so that turned out to be really great. So since about 2018, we've had this excellent funding. We've had this really strong team, multiple faculty members, lots of students, lots of postdocs, some staff. And we've done many, many things, but 2 things particularly I want to talk about. One is on the idea of synthetic route planning. If you want to make a new drug molecule or a new dye molecule, and you know the molecule you want to make, but you don't know how to make it, if the computer can suggest the recipe of all the steps you should do, all the reactions and separations, that's like very helpful. And Connor, who's probably going to talk about this later, maybe we'll talk about that. And that MIT's efforts in that area are packaged up in a software package called ASCOS that can propose synthetic routes to make molecules, and it's good enough that the pharma companies are interested. So it's I don't know if it's not maybe as best as the best organic chemists in the world, but it's good enough that it's useful for them. And then another whole aspect of this is trying to predict the properties. If you start from a molecular structure, can you predict how that molecule would actually perform? And that's very important if you want the computer to decide what to do, because it has to be able to both propose structures, evaluate if they're a good idea, and then figure out how to make them, all those different steps. And I worked on this second part about the property prediction, and that has a software package called ChemProp, which has turned out to be super popular. Let me just talk about how we predict things. So those of you who are old timers like me from the 20th century will remember that there were correlations. And we had handbooks and stuff that had correlations in them. And there's a whole field of QSAR, quantitative structure activity relations. It was a big thing. Linear free energy relations. You probably remember this if you're old. And mostly what they were linear correlations where you find some aspect of the molecule or of the reaction and you have some number like its thermo property or its color or something, and then you try to rig up some linear relation between those things you can see in the molecule structure and the property you're measuring. And this worked fine, and we have a lot of great examples of this, and you've heard of some of them like Benson's methods, Hammett correlation. We teach that to all the students. There's quite a few of them, actually. But the downside of this is, I mean, a good thing and a bad thing about linear models. The good thing is you can get a unique solution a lot of times, and if you can't get a unique solution, we have good numerical diagnostics to tell what's going on. So linear is kind of nice, but the problem with linear is a lot of properties we care about are actually not linear in anything. So it's like very difficult to actually get a good correlation. You can do better and if you can use a nonlinear model, and even better if you don't need an expert human to identify what's important about the molecule, but just have the computer figure out what's important about the molecule. And so that idea is the learned fingerprint idea, where you have the molecule, you have a list of properties, but you don't define what the properties are. Instead, the computer just figures out properties of the graph of the molecule, the structure of the molecule. And then you correlate those with the properties. And if you use a neural net, it's very flexible, and so it can fit almost anything. So you can get really good fits to the properties. And we have a nice software that does this at ChemProp. And if you're interested in that, a couple journal references there. And it's free, and you can download it. And then once you have, if you can do that, you can do the same thing for pairs of molecules, for reactions, any kind of combinations of stuff. Now, the issue with this is you're getting very, very complicated correlations. If you have neural nets and you have this procedure to make the learned fingerprints, lots and lots of parameters in your model. And so therefore, you really need lots and lots of data. But this is the one thing we never have in the chemical sciences, is you don't have enough data. And so this is an example here. So in this particular case, my students were able to scrape the literature and find 10,145 different solutes and different solvents where people had actually experimentally measured the solvation-free energy. When you start putting this data to train a model, you can see the error in the model drops really rapidly to begin with, but then it kind of like peters out, and you need more and more and more data, and you get less and less return on all that data. And so the poor graduate student who's doing experiment 1000, 1001, 1002, 1003, they might feel like they're really not making much progress for the advancement of science. And also, once you get these gigantic data sets, the curation is a killer. It's really hard that you don't let any bad numbers slip into the data set, that you have all the molecules drawn correctly, all the details about that stuff. But anyway, people do it. So if you somehow have the patience to get this much data, or somebody gives it to you for free, then you're good. But the data scarcity is a problem. And it's even worse than you might think, because first of all, almost all the data is on molecules that are commercially available. So we understand sigma Aldrich space very well. And we have zero understanding of other kinds of molecules because nobody ever does experiments. If they have to synthesize it first, it's too much work. So the data is really clumpy because typically people are interested in a certain property of molecules only for a certain class of molecules. That was the class that they were interested in commercially, typically. Or maybe some graduate student's boss just said, please do 27 derivatives of the same functional group or something. And so the data has less information content than you think. So I have those 10,000 solute-solvent pairs, but actually independent information there might be 3,000 or something, because a lot of it's kind of repetitive. Lots of water, for example, is the solvent. You've got lots and lots of water, not too much of other solvents, for example. And then some people have high throughput equipment and can measure a lot of new data, and Klavs works on that kind of stuff. But that's kind of expensive and a lot of work, and the companies that really invest in that and have technicians running high throughput experiments typically want to keep their data secret. So there is data, but you can't get it. Now, historically, you would go to like Beilstein or other books in the library, and you can get lots of data. But the people who own the copyrights on that data have recently recognized, oh, this data is valuable, so therefore we're not going to let you have it, unless you pay us a lot of money and give us all the rights. So we had some trouble with that, and Connor might tell you some stories. So basically, you're out of luck. Unless you're willing to read all the journal papers yourself and by hand write down the numbers, you're really not going to be able to get the data. So you have a choice. You either have to create the data yourself somehow, or basically you should change to a different career. So in my prior life, I was a quantum chemist, and I knew how to do quantum chemistry calculations. I thought, well, maybe I can use quantum chemistry to add data. So for example, for solvation, you can do a quantum calculation of what the solvation free energy should be for a solute surrounded by a solvent. It's not that accurate, but you can do it. And it's kind of very systematic, actually. You get all the trends and stuff are actually kind of sensible, because the quantum is like it's in absolute error, but the relative calculations aren't that bad. So you can do that. And then that's a way to get a lot of data, because the supercomputers have gotten really great. And so you can really crank through these calculations and get thousands or even millions of examples of data like this. And then what we did was we would train a model on this quantum data, and then say that we assume that this is good enough to do the learned fingerprints, to figure out which aspects of the molecules are important. We would lock those parameters, and then we would take our limited experimental data we could get and try to tweak this model to try to match the other model. So some people call this fine-tuning or like calibration to the experiments. And in cases where the quantum is pretty good to start with, and you have a lot of quantum data, this is actually a pretty good way to go. And so we have several examples where that worked. And so you can see how it worked for the salvation free energy case. Is if you only use the experimental data, you get a curve like this. But if you start from the quantum and then you just use the experimental data to tweak it, to fine tune it, you're already pretty good from the quantum. And a little bit of tweaking, you get really good. And so you don't need very much data. So you only need 200 examples is enough to get the same quality of prediction as you got from more than 3,000 data experimental. And you can also see a lot of funny things. In this case, the model is actually better than it appears because the model gets below the noise limit of the experiment. So we can't really test the model well enough to see exactly how accurate it is. But even this great model, has a few points that are way off. And this is a problem with all models, and is very obvious with machine learning, that you get almost all the points correct, and then some things are completely wacko. And this is very bad for chemical engineers, because if you base your design on that, you get the same problem. Just one last thing I mentioned. We try to predict reaction barriers. We could do nice fits and correlations. But you have to worry about physical things affecting you. In this case, the fact that the forward and reverse barriers are related by the heat of reaction is very important, and it causes a kind of data leakage. So our predictions look better than they really are because the model was actually learning delta H, which is a much easier thing to learn than reaction barriers. And we figured it out eventually and had to redo all our calculations to do better. And we still have some tail of reactions where we get very bad predictions, even though most of the reactions are predicted very accurately. I just want to acknowledge all the students and postdocs who did the work and all my collaborators from all the different groups involved in the groups that Klavs organized. And I just want to thank Klavs for getting me into this. I would never have gotten into this except he twisted my arm to join that project. And it's been a heck of a lot of fun, and I learned a lot. And I think both Klavs and I learned quite a lot from our students and from our colleagues. It was really pretty interesting. So thank you.
We have time for a question.
So it seems like this lack of data screams for something like an open source or something like what the genomic side of the community is doing. Is there a effort along those lines?
Yeah, and really you should ask maybe Connor about it, but there's an effort to try to get open data. I would say it's being fought against by Elsevier and some other people who own a lot of data and don't want it to be open. And I'd say the pharma companies are totally confused because they sort of want it to be open to help advance the science, but they also want to carefully protect their IP on their particular molecules. And so it's a quite complex situation right now.
Let's thank Professor Green again for a great talk. And our next speaker, I suppose Luke couldn't make it. We have Kashi, who's going to talk from On-Demand Pharma. And I actually have a loose connection, I guess, to that company. I'm actually on sabbatical right now at Dow. So I'm technically NYU and Dow. And I'm working with Anne-Catherine Bedard, who was in Tim Jamison's lab when she was trying to, I guess, take what Andrea did to another level that eventually turned into what Luke is working on.
All right, so hello, my name is Kosi. So I did my PhD with Klavs, but I also overlap with Luke and we're currently colleagues, and he couldn't make it last minute, so he messaged me on Thursday. He was like, hey, can't make it, can you do this talk for me? It's like, well, okay, but I'm not Luke. If you guys have met Luke, he's very Irish and he's very jovial. So I was like, well, I need help on the jovial front because I think I can kind of match the other part. He wanted me to tell him this joke about the Irishman dilemma, but I was like, I don't think I can pull it off, so I'll just do my best here. I think before going into details, we definitely do want to say thank you because I think this project, just a lot of people throughout the years, as you've kind of heard from the various presenters like Andrea, and it's been referenced today, but a lot of people have been on these projects and had to get all their names, make sure everything was spelled correctly. But with that, we do want to thank the man who made it all possible, Brian Smith from EHS. So he was definitely very crucial to the execution of the project. Klaus also helped. Klaus is there. So we do want to make sure gets the proper thanks there. So I tried to look back to find the initial call for proposals, but I saw this archived site for what this was, Pharmacy on Demand. So Battlefield Medicine, if you guys know the person who was the project owner, he was very interested in this problem of trying to solve, if you're for the Defense Department, when they're in remote locations, how to get drugs there when you don't have access to, the storage and you're not able to carry everything. So one idea is to make it on demand. So that's overcoming the logistical obstacles, and then obviously miniaturizing it so you can it's more portable, and manufacturing the actual doses. So, and this is one of the key things that I guess everyone has kind of talked about today is this multidisciplinary team in order to accomplish this goal. So in this case, you had Tim from chemistry, Alan Meyerson that's working on the purification and the formulation, and Klaus on the engineering side. And the idea here was, coming up with this generalizable scheme that you can then use to fit a lot of APIs into. So in this case, there's about 6 bays here, at least for the synthesis side. And then for the different APIs, you can mix and match or skip certain bays, different size reactors. And with that, you can generate, a whole host of APIs. So just going through the timeline here, at first you had this first phase, so this was back in 2016, and the real accomplishment here is this continuous end-to-end system in this fridge size form factor. You can do synthesis and purification in this system at about 100 to 1,000 of doses per day for oral solids and liquid formulations. And then obviously it's plug and play, reconfigurable. And then for the next phase, you had you had 2 units now. So you had one for synthesis and purification. And then you had also additional APIs that you can fit in there. Into the demo phase, and this is where I think a lot of Luke's work was, there's a 3-week campaign that they had to kind of simulate more of an actual production. So you had to do changeovers, and you also had to target about 1,000 doses a day. So in here, you can kind of see it's a little bit small, but relative to the other phases, they were able to increase the output for a whole host of them. Cipro was still a little bit low, but for most of them, they were able to get at least 1,000 doses a day. I think Andrea showed one of these pictures, but the whole floor was covered with these units. You see you have several students and researchers working on the various units. And this was specifically for Cipro production, and you have the tableter there in the top right-hand corner. But this was kind of the demo phase of the Pharmacy on Demand project. Later on, in the later years, the next goal was to do a more GMP Cipro production. And so what this got you is, gearing it towards commercialization. So you had to implement a lot more feedback control, 4x throughput increase. And this kind of just went on to further process improvements. Connected to this, so this was kind of the project that I worked on a little bit. There's another funding that money was made available to work on this molecules on demand. Now there have been versions of this since that's kind of improved on this idea of like a synthesis companion for a chemist for a lab scale production. And it's a similar concept where you have these bays that you can mix and match things as you need them. With reactors, with these small form factors. I think Andrea showed this image. And in this case, we had optimization. And what it was trying to show, like if you kind of integrate these things together, you can like take these simple examples of like chemistries, optimize, change out substrates, things that kind of transfer over. You can see some of the yields from like one optimization can transfer over for similar substrates, and if they don't, you just re-optimize for the same unit. So that was all throughout Klaus's time, and I think some of the learnings with the multidisciplinary kind of how do you solve these technical challenges we've tried to carry on. So since call this kind of getting the band back together. Tyler McQuaid, he was the program manager, currently boss of mine. But it was interesting about Tyler. When I was here, you hear Klavs because Tyler was this voice on the phones during like the updates. But Klavs had a lot of positive things to say. He's a chemist, but he's also very good at engineering. Like he kind of can ask these questions that are very quite detailed that I didn't think a chemist will be. No offense, but he was very good. I'll just stop right there. But going in and working with Tyler, it was just the opposite. So like he started like saying, oh yeah, Klaus, you guys were so good back there, all these positive things. And it was kind of interesting trying to see that like 2 equally very successful, very smart individuals have very similar high opinions of each other. It made me even further realize all the, how good it was working with you, Klaus. But we also had Naomi, who worked on the purification, is currently one of us, and Justin is here too. He worked in Tim Jameson, and Luke worked with us in Klaus's group, me. So one of the things we learned is how do you solve problems. I think Luke learned one thing here, and I think he's kind of carried it on, which is when you come into a challenge, have you tried heating it? And, it's something that he applies over and over to every problem he comes across, regardless of the application. But in one case, it did work. In in this case, we had the goal of making midazolam. And for this last decarboxylation step, it's usually hard to get rid of this isomidazolam that's generated with it. It's an impurity. You gotta make sure you make it in very small quantities. It's hard to purify. So this has been a challenge, and it's something that we came across too. So the question was, how do you get rid of it? Well, Luke's like, well, have you tried heating it? You generally do make this at a high temperature, but not this high. In this case, you have a slurry of the reaction mixture. So we were able to heat it, and then really not have to deal with isomidazolam at all. In order to do that, though, we had to apply some engineering principles. We made a hot plate reactor that you feed in at the very top here. You feed in your mixture of the acid with carboxylated midazolam. Feed it in. It gets heated to 230 degrees, and then it melts. And how hot you heat it also controls the flow rate. It melts, and it gets collected there at the bottom. We were able to collect a lot of very pure midazolam through this process. And here's just an image from the paper here that kind of describes that operation. And then with further downstream processing, just for the cleanup step. But you can see with the fragment collection, we're able to get an LCAP around 98%. And as you can see, we keep all the impurities low. So I think that's primarily all we wanted to cover here, and just wanted to end by saying very thank you to Klavs. Luke and I think one day we went boxing a few times, so that's fun. But here's an Irish author, gotta have an Irish author, with a quote here, ever tried, ever failed, no matter, try again, fail better. And I think that's a concept that really stuck with Luke, but also stuck with me as well. Just this concept of continuing to try and figuring out new ways to kind of explore whatever problem you're dealing with. And I think you guys may have gotten an email to submit names, so Luke was the one that kind of spearheaded this. But you can see some of the big words here for Klavs, like kind, supportive, patient, insightful, and that's something I think that resonates with a lot of us. So just want to say thank you, Klavs.
We have time for questions for Kosi. Ben.
Yes, I was always intrigued with the mobile platforms that produce the battle medicine. How often would that need to be restocked with raw chemicals? And also, how much waste and how benign or not benign is the waste that's continuously generated?
That's a very good question. And I think that kind of gets into the challenge when you start going into commercialization. So depending on your synthesis, your stock could be very, and the amount of APIs you want to generate, the stock solutions you have could be very diverse. You have to start with various precursors. I think the general, I would say long-term vision is if you can have a small subset of key stock that can allow you to explore a wide range of API, that would be the vision of that. And then with that will come the actual chemistry. If you can make sure your byproducts you're doing more solvent-free systems, all these things. So it's you get to the challenge of what is the limitation of production. I think the idea we're trying to look at now is maybe you don't want to distribute the final steps of the synthesis. Maybe what you're distributing is the formulation. So the formulation part is more distributed. So when you get to practical aspects, you might have to figure out how do you actually distribute things properly to get to that.
So for the 4 APIs, the first generation rig, the 4 APIs, how many separate stock solutions needed to drive that library? Is it on the order of 10? Is it the order of 4, 20?
I would say closer to
it's so dependent on the synthesis. Yeah. But it is an issue where one of the things that Luke did when he worked on the POD, on the demonstration, like to make 7 different APIs in a short period of time. The Cambridge Fire Department and the building rules, actually the logistics of bringing food and chemicals in the lab were immense.
Yeah, the brine aspect.
I wonder when anyone has that picture of all the bottles lined up along the wall, but there we go. That is an issue, right?
Other questions? Yeah.
How do you handle cleaning? Having worked in pharma for a while, cleaning is oftentimes more sophisticated than the actual synthesis.
Yeah. No, I think that's something we're experiencing now. So in the small scale, trying to get an FDA process for this small unit, you run into those challenges, changeover, clean in, making sure there's no carryover. That gets very challenging with the tiny units, trying to make sure you get everywhere and all that stuff. And you have to, that's where you start thinking of like, okay, what is it the problem we're trying to solve? Do we want to get it super tiny now, or do we want to address the kind of drug distribution issue? So it is a challenge. And I think right now we've kind of scaled up back a little bit. So
Thank you, Cozy. It amazes me where things have gone since the Novartis MIT Center, Klaus. Our next speaker is Connor Coley. The first time I met Connor was actually at an AICHE annual meeting on the sidewalk, and he looked stressed out. He was talking about all the work that he had to do because he was working on a DARPA project. And I thought to myself, wow, I don't know if I should write a grant to DARPA. But then several months later, I started reading these papers that were coming out, and I realized that they were defining a new field in reaction engineering.
It's quite an introduction to live up to. Well, it's fun to be here and help sort of close out this really fun day. Like someone has said before, it's a family reunion. It's everyone getting together and getting to see and celebrate the work that Klaus has created and sort of propagated in the field. So I'll talk a little bit about what drew me to Klaus's group and how that sort of evolved, and a lot of the themes that attracted me into this field are things that persist in the research that we do even today. So I liked this idea of efficiency, right? The idea of efficiency in experiments where you can be really creative and intentional about the way that you run experiments, whether they're reactions or testing molecules, to get the most information out of the least effort possible, right? And so if you go back and you look at the statistical design of experiments techniques, you'll start to see things like one factor at a time, where you just do this very simple screen to understand how x1 and x2 affect your process outputs, factorial designs, Box-Benken designs, et cetera. But then there was this also a notion that you could not predetermine necessarily this set of experiments that you would run, but adapt, right? And so you could end up with these sort of nice loops between some design space that you're trying to learn within, some sort of model, maybe it's a response surface or other type of machine learning model, and you could use that to help you select those experiments. And so when I was a grad student, first year grad student in the department looking at groups, one of the projects that I was super excited about was what Brandon Reisman had been working on. He was a 5th year PhD student at the time. He was working on these sort of self-optimizing reactors, as Steve Buchwald alluded to before. This is the paper that he mentioned at the top right. I'd seen also the work from John McMullen on these sort of self-optimizing reactors. And these are really cool because it's the notion that you can just set up a reactor and let it go, and it will find the conditions it needs to maximize its yield, right? It's sort of automated process development, at least for one step at a time, and then later extended through the Molecules on Demand project to multi-step cascades. And so the question that this is really trying to answer, right, all of these platforms, is that given all the experiments that you could run and given all the information we've collected so far, which experiment should be run, right? That's what these self-optimizing systems were trying to answer. And so this is what originally sort of brought me into the group, but we started to think a little bit more broadly in this context of the Make It program, because quite often, right, we're working with systems that are not new to they may be new to the laboratory, but they're not new to the field, right? People in the literature have run hundreds of thousands of Suzuki couplings before, or Buchwald-Hartwig emanations, and so how do we use that information to also help us and speed things up even further? So we broadened the question to now say, well, given all of the information that we and others have gathered so far, what's the best thing we can do? And as Bill alluded to, around this sort of 2016 time when we were starting on this path, we started negotiating with data curators and publishers like Elsevier. Not the most enjoyable process, but probably one of the better reasons that a student can tell their advisor that they need to meet with general counsel. So that was a lot of time well spent in those meetings. So eventually we got the data before they realized it was too valuable to give it to us for free. And then we started learning from it, right? We started using this kind of literature data as the basis for a whole bunch of machine learning and statistical inference algorithms. And so also mentioned was sort of ASCOS and this idea that we can build models that help us plan synthetic routes and processes to molecules we'd like to make. We can train these on the literature and we can start to ask now these new questions. Given everything we know, what is the best pathway, the best synthetic pathway to access these new molecules of interest? With the kinds of tools that we've been building since, again, 2016 or so, we can sometimes rapidly get these hundreds or thousands of different pathway options for different small molecules of interest. So we end up with these sort of great big sort of networks that we then have to worry about navigating to figure out the real answer to this question, right? What is the best pathway to use? Now, the definition of best, right, is something that is subjective and case dependent. This is something that I emphasize a lot teaching 1034, right? We have to say, what does best mean? And that sort of led us to a whole bunch of other projects and discussions around pathway optimality, right? Can we measure the feasibility of a synthetic route? Can we anticipate and quantify the impurity profile and how hard it will be to separate out the product we like? There's flow compatibility is itself a consideration that you might think about trying to build in and quantify so that you can use that to rank your pathways. But really this has led us into this sort of endless list of very difficult problems in process chemistry that we continue to collaborate on today. Also, as mentioned, was the MLPDS consortium, right, this morning by Crystala and then also by Bill. MLPDS is really this fantastic initiative, right, that Klavs led at MIT, sort of founded and brought together, right, not only folks at MIT, right, to collaborate on these topics across departments, but also a pretty impressive, not comprehensive, but a pretty impressive subset of the pharmaceutical and chemical industry, right, the big players, not just here around Kendall Square, but also globally. And so all of these companies, right, saw the leadership and saw the change that Klavs was leading in the field and trusted Klavs to really carry out this vision and be the consortium that they wanted to invest in and participate in and learn from. So it's been a really fun position to be part of the consortium, first as a student, now as a faculty member, and as a new faculty member especially, I will say the funding aspect is quite good as well. So that's been appreciated. Now in my research group, we've tried to continue some of these themes of experimental design in other contexts. So we don't just worry about designing synthetic pathways, we think about designing molecular structures and saying, given all the molecules we could possibly test, which ones should we test first, right? What are the highest priority ones? We can adapt techniques from Bayesian optimization to answer these questions, just like we use it for yield optimization, where we build models that help us navigate chemical space and choose what molecular structures look most promising and which ones probably aren't worth testing. So this model-guided optimization is also how we address this experimental design question for molecular spaces, and it ends up working empirically quite well, and we can use models to help us explore very large spaces of hundreds of millions or billions of candidate structures with orders of magnitude lower computational cost than a brute force search. Now, for molecular structures as well, we sort of run into this question of, not all molecules are created equal, and if we're just choosing which ones we should use in the lab, we should probably consider the synthetic cost as well, especially if we're talking about using robotic systems, like on the most recent sort of DARPA programs to carry out these experiments. We have to think about the actual cost to produce these structures. And so this notion of experimental design can be extended. So now we're trying to do cost-aware experimental design, and we can say, well, we have some set of structures that we could possibly test. We have some notion of their properties using tools like ChemProp that Bill mentioned, But we can also find synthetic pathways for each structure and then pose this as an optimization problem to down select the most informative yet synthetically tractable compounds for us to test. And so we're trying to bring this sort of extra level of formality to how we think about driving design cycles in medicinal chemistry and other settings. And as a sort of a test case, what better thing to do than piggyback off of another DARPA program that Klaus is leading, the AMD program, the most recent one. And so we can do things like reanalyze this design cycle that they actually faced experimentally, where they came up with 121 candidate molecules, right, that looked like they might have promising optical properties for this goal of creating new dye molecules. And we can pose this formal optimization and try to down select what are the structures worth making if I take into consideration their synthetic cost. And we end up with these small little networks where you can see, and here all the pink molecules are ones that we think are worth testing. And you'll notice that molecules that are worth testing, we've sort of strategically placed as intermediates in the synthesis of other molecules worth testing. So we're trying to be very economical with how we spend our efforts to get the most information out. And so this sort of the technical part of this, right, really is trying to build on these notions of experimental design that's maybe started, in my mind, with reaction optimization in Klaus's group, to more sort of general questions about how we think about scientific discovery and extrapolating beyond what we know to what we don't know. Right? We use models to help us do that extrapolation in a guided fashion. They help us quantify our uncertainty. We model the world. We let it tell us how good we think the new molecules we're going to make are going to be. And really, in this last point is when you think about discovery, you have to take some risks in what you're willing to run in order to actually sort of push the frontiers of knowledge. Now, just in the last few minutes, I want to sort of pivot momentarily. This is basically a slide I adapted from a 1034 lecture. When we teach experimental design and concepts of optimal experimentation, we usually start off with simplified systems. So we'll think about analyzing the case, where you have a linear model, you have some design matrix X, you've got some homoscedastic measurement error. It's a fun vocab word if you haven't heard it before. And analytically, when we have this setting, we know that the sort of parameter covariance matrix is a function just of the design matrix and not of the output variable. So you can basically say how you want to optimize the experiments you choose to maximize this information. Now, this analysis lets you define a lot of different optimality criteria. And so this is why it's sort of called this alphabet soup. So the optimal experimental design says I want to pick the experiments that minimize the trace of that matrix. D-optimality is that I want to minimize the determinant of that parameter covariance matrix. You have E-optimality, which is sort of figuring out the worst case scenario. V-optimality, defining a region that you want to be most accurate over. G-optimality, trying to minimize uncertainty in the maximal point in your design space. And so then naturally, I've been trying to think, what is KFJ optimality in this, right? What is the criteria, right, that we can use to optimally select what experiments we're running and how we're spending our time? So the key question in sort of KFJ optimal design is given all the ways we can spend our time, right, all the projects we can work on, all the collaborators we can choose from, all the students we can work with, what do we do? So I started writing out some list of things, and this is resonating with what we've heard earlier today. I started feeling like I was writing a LinkedIn post sort of partway through this. And I think really it's in looking at how I've perceived Klaus to run the group and shape his career in the field around him. This openness to new fields, right, has been a pervasive theme today, right? So let your interests take you in different directions, remain very open to changes in priorities and interests. Building communities, right, convening experts, collaborating with people within and outside of the institute. Another thing is, despite how many science papers Klaus has, he does believe that you should publish for the readership and not for the impact factor. So that's very much a message that he's consistently conveyed, right? We're not doing science for the headlines, we're doing science for the science itself and for the impact it can have on people's lives. I think there's this element of making sure that everyone knows that they're valued. For me, that involved things like letting me present some of the work that we were doing at these DARPA update meetings, just making sure that people know that they're a priority. Last thing from lab cleanout days, never throw away the mass flow controllers, even if it's been a decade since we've actually used a mass flow controller in lab. So no such thing as too many. So I was going back through just some old emails with Klaus, and I'll wrap up in just a second here. To sort of find, sort of our earliest communications and some emails from then. So this was when he was stepping down from department heads. So the first thing that he noted was, I'll be stepping down as department head this summer, and the main thing he's saying is that he wants to spend more time with the group and turn around papers faster. So really sort of letting people know that his priorities are shifting back to the research lab, and I think we all felt that. And then I came across the very first email I sent to Klaus. So I guess I was the first year I'd been talking to Brandon Reisman on the 5th floor of Building 66 about the projects, trying to get some background information before my first meeting with Klaus, which, by the way, the deafening silence comment is very real. And so I looked back at this email, and so I was, Professor Jensen, we met briefly, can I set up a time to talk? And I'd be delighted to meet. Alina can help with the scheduling, so thank you, Alina, for always helping with the scheduling. But what was sort of funny about this is that I had sent this email apparently on a Tuesday night at 10:13 p.m. And of course, Klaus replied 12 minutes later on the Tuesday evening because he was still working, still attentive. And even though I wasn't part of the group, I already knew that I was valued and prioritized. So thank you, Klaus, for all of your mentorship, your supports, your collaboration, for helping define the field, doing what you do to make this today possible, and seeing all the fruits of your efforts. And I don't think any of us know how to emulate KFJ optimality, but hopefully we're all going to try. So thank you.
Questions for Connor? Yeah.
Very impressive talk, thank you.
Can you comment if the approach can be extended to inorganic or organometallic type of molecules, synthesis of those, for which data is even lacking, right, more lacking?
Yeah, so in the inorganic synthesis space, I think there's a couple of issues. So one is that the data is less mature, right? We've, despite Elsevier's quirks, right, they and Chemical Abstracts have spent decades and decades curating these databases that we can learn from, and you don't see that in inorganic materials. But the other main difference is that organic synthesis has these locally stable states and isolable intermediates that lets us decompose the task in a very nice way into the sort of recursive process of retrosynthetic analysis. And for inorganics, you tend to not have those isolable intermediates, and so there are pretty substantial differences in how we have to think about representing synthetic processes when we port these tools over. But there are people trying and efforts on the way, but it's a very different problem, it turns out.
Any other questions for Connor? Yeah, David.
I know nothing about this, so I have a question. So you didn't mention anything about catalysis. I mean, is it the way that these reactions are done? They're so sort of standard, just pick a few things, or is there a huge number of different parameters that you have to choose from in the synthesis process?
Yeah, so the notion of sort of filling in the conditions once you've sort of sketched out what you might want your overall pathway to look like, that is its own problem. And certainly there's some reactions for which off the shelf, you open the cupboard and you pick the 12 most popular catalysts and something works. There's a lot of reactions that are like that, thanks to folks like Steve. Certainly for other chemistries, that is where the bottleneck is, right? So you might think that some pair of substrates is compatible with this reaction type, and you've designed it in this very clever way, but then you might not be able to find the right catalyst ligand system to achieve it. And so there are cases where that happens, but for a lot of the sort of simpler medicinal chemistry type pathways, you can usually find something. Yeah.
Thanks again, Connor.
Thanks.
Now I'd like to invite Klaus to make some remarks.
So, first of all, I'm deeply honored. I'm very appreciative of all the wonderful things that you've said, and most of all, for the fact that you actually came here to this family reunion, to this group reunion. I think that's really wonderful. And I was hoping that if we did this, and some of you know that I was sort of debating whether this should even take place, and some of you were fortunate were very nice in helping me get through that decision process, saying that they thought that would be good. It's really been wonderful to see you all together and interacting. We put in time in the schedule for that to happen. It's great to see it happening, and it's been terrific to actually see you all. And then for a select few, we couldn't fit everybody into the schedule. I'm sure that many of you could have given equally interesting and motivating talks about what you're doing. I mean, you've all had amazing careers and you've done incredibly well in what you're doing. And it's very rewarding to see how successful you've been and how many different things that you have moved into and managed to accomplish. I was fortunate enough that I got hired at Minnesota. I had really great mentors. My office was right across the door from the hall from my reaction engineering idol, Gus Aris. And I always thought that, I like to be like Gus, really know everything there is to know about reaction engineering and how to apply it, and do it quietly and nicely, and not make a big fuss. So that's sort of been one of the idols that I've had. And some of you say I've been trying to be temperate. I think it comes from really having admired how Gus did this. There were other people in the department that had very different ways of doing things that made it, some of you from Minnesota will remember, that made it a very exciting place to work. I already at that days, I learned a lot from actually kind of branching out from what we normally did. So I often would go to what was known as the low tension seminar that Skip Scriven and Ted Davis ran, because it dealt with low tension fluids, but actually was a really great training ground if you wanted to know about fluid dynamics and transport and porous media and all those things. And low tension is maybe not the right characterization. But there was a lot of things to learn, and that actually led to a lot of the research project, like Raj's thesis project really grew out of the percolation theory that I learned from that particular interaction. And then this idea about how did you get into electronic materials? Well, part of it was sort of an offhand comment from Lenny Schmidt, who said, "Hey, have you ever heard about this stuff, CVD? It sounds interesting." And I happened to spend the summer working at Exxon, and I did actually, you shouldn't tell them this, but I did spend a few afternoons at Bell Laboratories, which was not too far away from Exxon Research Laboratories at that time, trying to figure out how things actually work. And then I made some contacts at Bell Laboratories. And I think it's really important in whatever problem one gets into, you really have to understand what's the problem? What is there actually here that you could solve and make an impact? Because it's obvious that there are lots of problems where you can take sort of chemical engineering and apply it and get a nice model out of it, but is anyone going to care? But here it was clear that there were actually a lot of very clever electrical engineers that were running chemical processes or desperately trying to figure out how they actually work. Because if they knew how they work, they could make them better. And so that's actually how, a lot of the projects that you heard about from some of the members of the group, both from Minnesota and also from MIT, that's sort of how the origin of those programs, and then working with very clever engineers from those fields that really knew the fundamentals. And in those days, going and giving a talk at Bell Laboratories was sort of like doing a preliminary exam. You really had to be on your toes. There were clever people in that. Some of you may know Frank Bates. He's quieted down since it was at Bell Laboratories, but there was different in an academic laboratory, academic talk. If you give an academic talk, they give you 50 minutes. There you were about 10 words into it, and there someone would say, "Are you sure?" or worse things. So anyway, I think that was a very good training ground to really focus on the fundamentals and learning people around you that sort of emphasize that. And it was a very collegial department. And I had really no reason to leave it, but I'm really grateful to Bob Brown, and who's not here, and to my colleagues at MIT for attracting me to MIT, because that was like walking into the candy store. It was just terrific. It was a very also a very collegial department, but it was most of all, it was also a very collegial, multidisciplinary operation. People talk about how do you do multidisciplinary research. I think one of the things that works at MIT is you don't actually have to go outside to talk to your colleagues. It's actually in many ways closer for me to go and talk to Steve Buchwald than actually going to talk to Bill Green, because with Bill Green I actually have to go through the tunnel or I have to go outside, right? But whereas Steve, I go down one floor and around the corner. So it's and so that sort of collegial collaborative atmosphere led to lots of collaborations. We had a lot of collaborations with Mundi Bawendi on the synthesis of quantum dots that grew out of that. We had interactions with Marty Schmidt. We could never have done these microreactors had it not been for Marty's superb knowledge of microfabrication. I mean, the heights that Alex and Samir and Lionel took making microreactors after Ravi had actually figured out how we could make the first one, right? Would never had happened had it not been for Marty's knowledge of all of that. And it's sort of, these interactions really have continued when we got into doing chemistry. The people that know chemistry, right? So, I mean, one of the nice things that the Novartis project did was really to bring us in close contact with Tim Jameson and Steve Buchwald, and really begin to understand what's really important, what kind of molecule should you do, right? You know, Ryan can attest to the Steve Buchwald laugh test, right? So if you now do a study, are the organic chemists going to laugh because it's so simple, or they're actually going to think it's an interesting problem, right? And I think that's something that you have to think about if you're doing multidisciplinary research. You have to satisfy both parties in a sort of, the machine learning and the ML PDS have come up a couple of times. I want to really emphasize that it really was a very joint effort. There was a call for proposals for DARPA that called specifically for, as Connor said, can we learn from all the prior information in chemistry? And at that point, I had been department head and spent, as Chris can, many Monday mornings in the dean's office. But one of the advantages I had out of that was I actually had a network of people I could talk to about who's doing what. And also, you get, when you see everybody's tenure cases coming through the system, you get a sense of what goes on in the system. So I went to the head of computer science and I said, we have this opportunity. Do you know anyone that's willing to talk to an engineer or actually get engaged in this kind of a problem? And he said, talk to Regina. And Regina actually was open enough to think, hey, this looks kind of interesting, let's talk. And so we spent a lot of time talking about what could possibly be done. And then, of course, it was helped immensely that Connor and Wing Jin was sort of a student of a, you know, as Bill said, the students talking together. And, but that's sort of how it started. And Tommy Yakala was Regina's partner in many ways in the research that they did. They had a very tech collaboration. So when we started working with Regina, Tommy came along with, brought a lot of technical knowledge along with it. Bill sort of gave you a story about our interactions. I'm actually grateful to Bill for many things, but first of all, we finally managed to collaborate because I was doing quantum chemistry calculations for organometallic compounds for doing CBD, and I knew that Bill was doing all this really interesting organic chemistry for combustion and so on, with quantum chemistry simulation and kinetic simulation and automatic reaction generator. And there ought to be some way that we could use some of all those tools. And so this was a really great way that we got together. The second reason I'm really grateful for Bill is that he was willing to be the executive officer at the most difficult point. And because as those that knew their MIT local maybe know, is that the executive officer has these, this wonderful opportunity to do the two most desirable jobs for a department head: planning, teaching, and organizing space. and this business of organizing space when you're doing a renovation becomes just a nightmare. And Bill did this superbly. And this was not helped by, we had several people from MIT physical plant that had sort of ideas of how you open valves to pipes that have no outlet. So we got the building flooded multiple times. I think we had a club of the flooded. But everybody took it sort of in that spirit, I think in part to Bill's very good humor approach to this whole problem. Plus, you didn't have to find space for people once. You had to find space for people every 6 months because we had to do this floor-wise. And so, and then we also renovated across the street. So it was a kind of a moving picture. And as Yiming alluded to, then besides that, there were also this issue of the money and finding people that were willing to actually pay for this. So there was it was an interesting period, and then trying to keep the research going. But I really want to thank you, the students and the postdoc in that group, because it's really been wonderful to see you again. I mean, this is the greatest pleasure is to work with you, because I mean, it's from when you first started in the group to when you become the expert on telling me, this is how it is, and when you graduate with your thesis, and then seeing you doing really well and actually moving into new areas and conquering, that's been terrific. And you, many times people have said, I took a chance on you. I actually don't think I did. A lot of times, in your case, Ryan, I actually valued the fact that you actually had some industrial experience. I thought it would be helpful. For the rest of us. And in many cases, Andrea brought some very deep engineering knowledge and skills that we really would be very helpful on the fluidic side, and obviously paid. And so in each case, there were things like that made sense. And so, you have, talking about trying new things and so on, I think a lot of the new project we did was because you actually tried those new projects. And I remember, when people sort of was branching out into, to me it sounded really interesting and curious. And so I was curious about the answer too. And then the second question was, all right, so now if we do that, how am I going to fund that? Or do you think I can justify this under this contract? So, which, I'm sure that any of you that run in research groups are familiar with having these kinds of challenges. And so, but I think it was always worthwhile because, it was a new set. And so I want to really thank you because it's been, and it's going to be the most difficult thing about stopping is actually, you're not having this sort of interaction on a daily basis with a group of very energized people that want to do something fun. But so as you know, I've gone post-tenure, which is this nice MIT way that will allow me to continue to get involved, stay involved in research. And also I can teach if I volunteer and if there's an opportunity. And I'm certainly also willing to help the department in that fashion. So, but ultimately, it means that I get more time. And that's actually, I think, very desirable because there's also other things I can do with that time. And I plan to spend that time I have for many years done a very bad job of playing the piano. I sort of reserved part of my Saturday to play piano, which was what Hong Lu was sort of referring to, because that was the only time I think I could find. But it was not very much, and you don't make much progress under those circumstances. And I actually right now have a wonderful 88-year-old teacher who's can assign me more music and more challenges than I can imagine having time for, even if I completely stopped. So I think I don't have to worry about being bored. But I also used to do other things. I was very interested in art. And actually, so when Felice came around and was interested in images, I thought that was wonderful. And as many of the people in the group will remember, I was kind of always picky on some of the slides. And so I understood what you said, and I really appreciated the collaborations that we've had on that, because I think it's very important how you communicate and make sure your figures are visual and actually have the right message, and also are honest in terms of what they present. And so I'd love to do more of that. And then there are a couple of other things I wanted to mention too. In terms of the schedule today, we couldn't put everything in here. But in what you haven't heard about are from the people who are still actually in the group and working. And well, Ben just graduated in January, but Bren Kosher and Matt McDonald were part of the paper that, the work that Bill was referring to that was published in Science here in January, and continuing to work on how to use these automated systems for discovery. And Shinji Lee is here and working on process chemistry issues and some of the things that Connor alluded to in terms of, is there such a thing as a perfect pathway? And then SK is working on a multitude of machine learning problems having to do with yield prediction, understanding large data sets, and thinking about how do we actually predict impurities. And so there's still a lot of interesting things going on in the group. And I should mention Zach, who is actually taking us to new heights in terms of the machine learning, thinking about what else can we, how can we otherwise interact with machine learning tools. We have worked in the past on Andrew Zart, who's not here, did a nice work on using machine learning to come up with new reactions for doing electroorganic chemistry. And we've been looking at electroorganic oxidation reactions. Can you predict whether a molecule can be oxidized by electrochemistry? And can you predict the site selectivity of that? And Zach has told us how much you actually can do with large language models, which is quite impressive. You can screen the literature that when I started looking at how to find out what's CVD and what do you do with CVD, I went, which most of you have never tried, I think, went to a card catalog, right? And chemical abstracts in book form, and put it all on index cards, right? Took forever. Now, fortunately, now you can put it into Google or whatever search engines you use, or better search engines. The next step was, when you download these articles, you give them to ChatGPT-4 and you ask it to read it and tell you what are the reaction conditions and so on. You very quickly get all this tabular data of what you can use. And then writing code. Many of you wrote long codes in Fortran, C, later on Python, right? A lot of it was hours spent debugging and trying to figure out the work. Well, at least simple codes, you can actually use machine learning, large language models to basically write your Python code. And so I think things are going to change rapidly in the next 10, 20 years in terms of how we actually work and interact with all these tools to kind of improve our working environment. And I think it's going to be fun because you're going to have all this information and you can really think about what problem do I want to solve and how to solve it. And then it becomes easier to organize the information than what's known before. So there's less of a chance of actually repeating what other people have done in the past. And so, I would be, there are a lot of people I would like to thank. As I said, first of all, you members of the group, former members of the group, and of course my colleagues, both back in Minnesota and here at MIT. And of course my family, who actually not always were so, you know, I've spent a lot of time in the lab. I spent a lot of time with you, and it meant I spent less time with them. And so I would like to thank Chris and my children for actually having given me the chance to do that and actually allowed me to do that. And Chris has been very generous in terms of actually, but so I also like to use some of my time to make up for that and spent a little more time with Chris and doing some of the things we actually enjoy doing together. And then finally, I think we should all thank Kathy and Gwen, particularly Kathy, who has been, as you know, the one that has been handling all the many tasks of this organization of this meeting, and then Gwen, who stepped in to help us today. But again, I want to thank you all for, you know, a wonderful career. Thank you very much.